Welcome to Deep Dive Lab, where we break down and dissect cutting-edge scientific research, from advanced biomedical technologies to breakthroughs in AI, material science, chemistry, and physics, all told as compelling, easy-to-follow stories.
And we have a truly wild one today.
We really do. So, I want you to imagine this. It is May 2026. You are on a luxury cruise ship, the MV Hondius.
Oh, nice.
Right. You've just sailed out of Argentina. The ocean is beautiful. The vacation is just picture perfect. And down in the main dining hall, there is this big birthday party happening.
People mingling, dancing, that kind of thing.
Exactly. People are passing drinks, laughing, just, enjoying the evening. But shortly after that party, passengers start falling ill. And I mean severely ill.
Yeah, and it begins with symptoms that are incredibly easy to dismiss at first.
Like what, just a cold?
More like a bad flu, extreme fatigue, severe muscle aches, maybe a spiking fever. But then, within a matter of hours, it just rapidly progresses.
Wow, that fast.
Yeah. Soon, people are literally gasping for air. Their oxygen levels just plummet, and their lungs actually begin filling with fluid.
Which is terrifying. The medical teams on board are absolutely scrambling at this point. They isolate the patients, they run the standard panels,
Right, checking for the usual suspects.
Right, and it's not influenza, it's not COVID. When the results finally come back, they are completely shocking. It's hantavirus.
Yeah. Specifically, the Andes strain.
Which brings us to the core of today's deep dive. We are exploring this microscopic mystery that suddenly became a very visible, very human crisis on that ship. Because, hantaviruses are mostly associated with dusty barns and rural rodents.
Right, sweeping up mouse droppings in an old shed.
Exactly. Not sipping a cocktail on a luxury cruise liner.
Which is exactly what makes that birthday party detail so chilling. The Andes strain is unique. It breaks that usual rodent-only rule and can actually spread from person to person.
Wait, really? Just from being near someone?
Yeah. Close contact at that party was enough. One person just passed it to another. Tragically, 3 people who were on that ship died, and several others ended up fighting for their lives in biocontainment units back in the US and Europe.
Man, it sounds like the opening scene of a Hollywood contagion thriller.
It really does.
But it brings us to a fascinating and honestly a really strange scientific question. This is the big mystery we're unpacking today. Here's a strange question. Why does a virus that typically lives quietly in mice, causing them absolutely no harm, by the way, suddenly cause human blood vessels to spring massive leaks without actually destroying the cells themselves?
That is the ultimate paradox here. It totally defies how we typically think viruses operate.
Right, because it's sort of like a burglar who doesn't even smash a window to get in, but somehow convinces your own home security system to flood the entire house.
That is a brilliant way to conceptualize it. And, scientists have actually recently uncovered the precise molecular lock this virus picks to get inside and trigger that flood.
Oh, wow, they actually found the lock.
They did. But to understand how they found it and, well, what it means for your own safety, we first kind of have to look at what the scientific community originally believed and why they got it wrong.
Okay, let's set the stage historically then. Because when we talk about hantaviruses, we are actually talking about 2 distinct medical nightmares depending on where you happen to live, right?
Exactly. We divide them geographically and medically into Old World and New World hantaviruses.
Okay, so let's start with the Old World. What does that look like?
So in Europe and Asia, you have the Old World strains. The most famous is the hantavirus. It was discovered back in 1978 near the Hantan River in South Korea.
Oh, so that's where the name comes from.
Right. But doctors first really encountered it during the Korean War in the early 1950s. Over 3,000 United Nations troops suddenly came down with this mysterious, really terrifying hemorrhagic fever.
Wait, so if the Old World strain was causing hemorrhage fever in soldiers over in Asia, what exactly were the New World strains doing to people in the Americas? Because the symptoms on that cruise ship sounded completely different.
They are completely different. The Old World viruses cause hemorrhagic fever with renal syndrome, or HFRS.
Renal, so kidneys.
Yeah, exactly. Yeah. As the name suggests, it primarily attacks the kidneys. You see massive drops in blood pressure, acute renal failure, internal bleeding, and it has a mortality rate of around 5 to 15%.
Which is bad enough. But the New World viruses?
They go straight for the respiratory system. Back in 1993, there was a sudden outbreak in the Four Corners region of the southwestern U.S.
Oh, I remember hearing about that. Young, healthy people were just suddenly getting sick.
Young, perfectly healthy adults were suddenly dying of rapid respiratory failure. Scientists isolated a new hantavirus there called the Spillover virus.
Syndrome, so the virus without a name.
Exactly. These New World viruses, including the Andes virus from our cruise ship, they cause Hantavirus Cardiopulmonary Syndrome, or HCPS,
And instead of the kidney shutting down, the lungs fill with fluid.
rapidly. It causes what we call non-cardiogenic pulmonary edema.
Okay, meaning it's not caused by heart failure.
Right. The fluid buildup is so fast and so severe that patients, they essentially drown from the inside. And the mortality rate for HCPS is staggering. It can reach up to 40%.
Wow, 40%. That is terrifying.
Yeah, it's one of the most lethal viruses we know of.
Okay, so whether it's the lungs in the Americas or the kidneys in Asia, the end result is basically the same,
Yeah.
right? Massive fluid leakage from the blood vessels.
Exactly.
And for years, scientists were trying to figure out how the virus was pulling this off. They were, looking for the doorway the virus used to enter our endothelial cells.
Right, the delicate cells that actually line the inside of our blood vessels.
Yeah. And for a long time, the established consensus was that the virus was using known cellular receptors, specifically proteins called beta-3 integrins, or another one called DF.
DF stands for decay accelerating factor. These are just proteins that naturally sit on the outside of your cells. And the belief was that the virus was just hijacking them to force its way inside.
But there was a major biological puzzle that just wasn't fitting together with that theory, because the hantavirus is what virologists call non-lytic. Can you explain what that actually means?
Sure, think about a severe influenza virus or like Ebola. When those viruses invade a cell, they replicate furiously, and then they literally burst the cell open to release the new viral particles.
Like blowing up a factory once you're done using it.
Exactly. They destroy the tissue. That is a hemolytic process. But hantavirus doesn't do that. It slips into the human endothelial cells, quietly replicates, and then just leaves the cell perfectly intact.
So there is no direct cell death caused by the virus itself,
None.
which makes the lethality so confusing. And to add to that confusion, when researchers in the lab tried to chemically block those beta-3 integrin or DF receptors, the supposed main doorways, the virus still found a way in.
Yeah, blocking them didn't fully stop the infection, which meant those receptors couldn't possibly be the whole story.
Okay, let's unpack this. If the front doors, those beta-3 integrins, were chemically blocked, but the virus was still flooding the house, they had to be missing a hidden back door.
They absolutely were.
And if the virus isn't blowing up the cells like a typical pathogen, why is it so deadly? I mean, what is actually causing the lungs to fill with fluid if the blood vessel cells are technically totally fine?
That is a phenomenal question, and the answer is that the virus isn't actually killing you. Your own immune system is.
Oh, man. That is the ultimate betrayal.
It really is. It's a process called host-mediated immune dysregulation.
Okay, big words. What does that actually look like?
So the virus acts as a silent trigger. It sort of tricks the body into this massive, catastrophic overreaction. When the virus infects those blood vessel cells, your immune system eventually notices something is wrong and releases a massive wave of pro-inflammatory cytokines.
Ah, the famous cytokine storm.
Right.
It's like pulling the fire alarm, but then sending in 1,000,000 fire trucks that just end up smashing the entire building to pieces, trying to put out a tiny trash can fire.
That is precisely it. But hantavirus does something even more insidious alongside that storm. It makes those blood vessel cells hyper-responsive to a protein called VEGF.
Vascular endothelial growth factor.
Correct. Normally, VEGF just helps regulate blood vessel permeability, but on top of that, the virus activates the kallikrein-kinin system in your blood, which releases a molecule called bradykinin.
Wait, hold on. Slow down. Kallikrein, what? You're throwing a lot of heavy chemical names at me all at once.
Yeah, fair point. Let's break that down. Imagine the cells lining your blood vessels are like individual bricks.
Okay, bricks, I'm with you.
And between those bricks, there is a strong biological mortar. It's a protein called VE-cadherin, and it holds them tightly together so your blood stays inside the pipes.
Makes sense. Bricks and mortar.
So when the virus triggers that VEGF hyperreaction, it forces the cells to sort of internalize that VE-cadherin. It literally pulls the mortar out from between the bricks.
Wow.
Yeah. And then that molecule I mentioned, bradykinin, sweeps in. Bradykin's a really potent inflammatory mediator. Its whole job is to further dissolve those tight seals.
So the cells themselves aren't dead. They've just been chemically tricked into unzipping from one another.
Exactly. The barrier entirely breaks down. Blood plasma just leaks right out of the vessels and pours into the surrounding tissue.
Which in the kidneys causes the renal failure, and in the lungs causes that fatal pulmonary edema.
You've got it.
That is both elegant and terrifying. The virus sets up the dominoes and lets our own immune system knock them down.
Basically, yeah.
So if the known doors are blocked and the virus is still getting in to trigger this unshipping, how did scientists figure out exactly how it was getting inside to pull that fire alarm?
Yeah.
I mean, this is where the story gets really interesting. How do you even begin to search a human cell for one microscopic, unidentified backdoor?
Well, you bring in modern genetic tools. Specifically, we have to look at a groundbreaking 2021 study. This was published in the journal eLife.
Okay, who's behind it?
A team of researchers, including Dr. Kartik Chandran and Dr. John Dye from USAM, the U.S. Army Medical Research Institute of Infectious Diseases.
The Army's top biodefense lab.
Exactly. They decided to crack this case using CRISPR-Cas9 genetic engineering.
The famous molecular scissors.
Yes. So they took a line of human microvascular endothelial cells. These are called time cells. And they're great because they perfectly mimic the tiny, really delicate blood vessels in our lungs that those New World viruses target.
Okay.
So the team did something brilliant. Using CRISPR, they systematically deleted candidate hantavirus receptors one by one.
They basically went door to door inside the cell, changing the locks one by one with CRISPR until the virus was finally locked out.
It's exactly like that. First, they deleted the genes for beta-3 integrin and DAF, the old suspects.
Did it work?
Nope. The New World viruses, Andes, and SIN number, they still got right in. Infection proceeded normally.
So those definitely weren't the master keys.
Right. But then they used CRISPR to snip out a receptor called protocadherin 1, or PCDH1.
PCDH1. What does that normally do when it's not, being hijacked by a lethal virus?
It's a protein that's normally involved in cell adhesion. It helps keep tissues together.
Okay, so what happened when they removed it?
It was a total wow moment in the lab. The human endothelial cells suddenly became highly resistant to the virus.
Wait, really? Just from that one protein?
Yes. The Andes and SIN number viruses were completely locked out.
That's incredible, just by removing that one specific lock. But, if they prove this in human cells in a petri dish, how do they know it works in a living, breathing respiratory system? A diff isn't a lung.
That is the exact hurdle they had to cross next, because you're right, it's not the same. So in hantavirus research, the gold standard animal model is the Syrian golden hamster.
Hamsters.
Yeah, because they actually develop a lung disease that is incredibly similar to human HCPS. So the researchers genetically engineered a group of hamsters to completely lack the PCDH1 receptor.
Okay.
Then they exposed both normal hamsters and the engineered hamsters to the deadly Andes virus.
The same exact strain that hit the cruise ship.
The very same. The normal control group succumbed to severe lung injury, as you would expect, but the hamsters, missing the PCDH1 receptor, they were completely resistant.
No way.
They survived without any lung fluid buildup at all.
Wow. That is an absolute confirmation. So they definitively proved that PCDH1 is the master key for these deadly lung-targeting hantaviruses.
They did. Finding that gives scientists a literal physical target on the cell surface to block, preventing the virus from ever initiating that fatal immune system overreaction.
But that only helps if you're already in a hospital bed with doctors who know how to block it. What does this all mean for you, the listener? Are we just waiting for the next cruise ship outbreak, or are we actually building new defenses?
Well, to answer that, we have to kind of zoom out from the microscope and look at the globe, because the environment itself is changing the rules of the game here.
How so?
Hantavirus is fundamentally an environmental disease. And this brings us to a concept ecologists call the bottom-up trophic cascade.
A trophic cascade?
Okay, break that down. It's all about climate change disrupting ecosystems. We are seeing more intense El Niño events, wildly altered rainfall patterns, milder winters, expanding urban heat islands.
But how does a shift in global weather patterns connect back to a virus in a mouse?
Let's say an area experiences unusually heavy rainfall due to an intense El Niño. That excess moisture causes what's known as a mast event.
A mast event?
Yeah, trees, shrubs, plants, they suddenly produce massive amounts of seeds, nuts, and vegetation.
An all-you-can-eat buffet for the local rodent population.
Precisely. The rodent population just absolutely booms. But then an extreme weather event hits the following season, maybe a prolonged drought or a severe flood. Their natural habitat is either depleted of water or completely destroyed. So where do these millions of newly born rodents go?
Oh, I see. They are driven into human environments seeking shelter and resources. Farms, suburbs, city garages.
Exactly. And as they move in, they leave behind droppings and urine.
Which dry up, turn into microscopic dust and get inhaled by people just, sweeping out their garage or opening up a summer cabin.
Climate change isn't just making the planet hotter, it's actively driving the virus carriers right to our doorsteps by destroying their equilibrium.
And as we saw with the Andes virus, once it makes that jump to humans, it occasionally figures out how to go from person to person.
Yeah, which is the scariest part.
But, I was reviewing the notes for today, and there's a recent study published in PLOS Pathogens that really caught my eye because it seems to rewrite this entire timeline. We keep talking about rodents, but they aren't the original hosts, are they?
They are not, and that discovery completely upended decades of virology. Yeah, researchers went looking outside the standard rodent populations and identified 4 completely novel hantaviruses.
With names like Huangpi and Longquan, right?
Exactly, and they found them in bats and shrews in China.
Wait, bats. We are dealing with another bat virus.
Bats and insectivores like moles. By mapping the genetic family tree of these viruses, a field called phylogenetic scientists realized that hantaviruses didn't actually originate in rodents at all.
Wow.
They likely originated in bats or insectivores millions of years ago. They've just been quietly co-evolving, adapting, and jumping between mammalian species ever since.
That shows an incredible, almost terrifying ability to adapt. I mean, if a virus can jump from a bat to a mouse to a human and then figure out how to hijack a highly specific protein like PCDH1, that is a versatile pathogen.
Highly versatile.
Which makes the question of treatment so urgent. If someone walks into a hospital today with a hantavirus infection, what do we actually have to fight it?
Currently, we have no FDA-approved vaccines and no targeted antivirals.
Yeah.
It's mostly just supportive care. Doctors provide oxygen, put patients on ventilators, and carefully manage their fluids. For the old-world kidney-targeting viruses, doctors sometimes use a broad antiviral called ribavirin.
Does it work?
It helps, but it has severe side effects. Like hemolytic anemia, it actually destroys your red blood cells. And unfortunately, clinical trials have shown that ribavirin is basically ineffective once the severe lung symptoms of the New World HCPS begin.
That's grim. So how do we use these new genetic maps, like the CRISPR discoveries, to fight back? Are we testing new drugs?
We are definitely entering a new era of countermeasures. For one, researchers are testing intelligent combinations of drugs. For instance, pairing ribavirin with favipiravir.
What does favipiravir do?
It's a broad-spectrum RNA inhibitor. It essentially jams the virus's genetic copying machine, and it has shown strong protective efficacy in animal models.
Okay, so attacking the virus's ability to replicate from multiple angles. But what about the fluid leak? Let's connect that back to the analogy used earlier. If the virus is the burglar and bradykinin is the chemical unzipping the bricks of our blood vessels, what can we do to stop the unzipping?
This is where host-directed therapies come in. Doctors are now testing a drug called icatibant.
Icatibant?
Yeah, it's a specialized bradykinin receptor blocker. So instead of trying to shoot the burglar, the virus icatibant is essentially super glue for the mortar.
Oh, that's clever.
Right. It tells the human immune system to stop unzipping the bricks. In a few case studies in Europe, administering icatibant dramatically stabilized the blood vessels and improved patient outcomes.
That is brilliant. Don't even fight the virus, just reinforce the walls while it's inside.
Exactly.
What about prevention, though? Are we looking at vaccines?
Vaccine innovation is accelerating rapidly. We aren't just relying on old-school inactivated viruses anymore. Researchers are developing next-generation DNA vaccines, VSV vectored vaccines that offer long-term protection from a single shot, and mRNA vaccines, similar to the technology used for COVID-19.
Actually, I saw a note about a preclinical mRNA vaccine out of the University of Bath using a technology called insilication. What exactly is that?
Oh, it's a fascinating breakthrough. One of the biggest problems with mRNA vaccines is that they require extreme cold storage, which is, incredibly difficult in the rural or remote areas where hantavirus typically strikes.
Right. Maintaining a cold chain in the middle of nowhere is almost impossible.
Exactly. So insilication involves essentially encasing the fragile vaccine proteins in a microscopic lattice of silica glass.
Glass.
Yeah, silica glass. This stabilizes the vaccine so it doesn't degrade at room temperature. It removes the need for refrigeration entirely.
Wow. That is massive for global public health deployment.
It truly is a game changer. And finally, building directly on the discovery of that PCDH1 receptor, scientists have successfully isolated monoclonal antibodies from human survivors of hantavirus.
Wait, so they can just use survivor antibodies?
Yeah, antibodies with designations like ADI 65534 and SAB 163. These are engineered to bind directly to the viral proteins or the PCDH1 receptor itself, physically blocking the virus from entering the cell.
Like giving the passengers on that cruise ship an immediate passive shield the moment an outbreak is detected.
Precisely. It's instant immunity.
Wow. Let's pull all these threads together because we've covered a lot. We started with a luxury cruise ship in 2026, dealing with a deadly, invisible stowaway. What are the core takeaways anyone listening today should walk away with?
I'd say there are three critical insights here. First, on a microscopic level, hantaviruses are master infiltrators. They use highly specific cellular locks like the newly discovered PCDH1 receptor to silently infiltrate human blood vessels. Second, the sheer lethality of the disease, that horrifying fluid buildup in the lungs or kidney failure, isn't from the virus directly destroying your cells. It's a tragic case of friendly fire.
The cytokine storm and the unzipping of the bricks?
Exactly. Our own immune system catastrophically overreacts, dissolving the seals between our blood vessel cells. And third, on a macro level, we absolutely cannot ignore the environment. Climate change, extreme weather events, and urban expansion are actively shifting the ecology of the animal hosts. We are blurring the boundaries between wild habitats and human spaces, bringing these ancient viruses right to our doorsteps.
It really highlights how interconnected everything is. A shift in ocean temperatures causes a tree to drop more seeds, which feeds a mouse, which migrates into a garage, carrying a virus that specifically evolved to pick a microscopic lock on a human blood vessel.
It's wild, isn't it? It's a perfect example of why science is a real-time, ongoing journey. Think about how human understanding has evolved here. We went from facing a mysterious, terrifying hemorrhagic fever during the Korean War in the 1950s, to tracing it to rodents, to uncovering its deep evolutionary history in bats, to dealing with person-to-person spread on a modern cruise ship.
Right.
And now we are literally using CRISPR to map the exact molecular entryway the virus uses. By understanding both the macroenvironment and the microcellular biology, we are finally predicting and preventing these crises rather than just reacting to them.
It's incredible progress, but, it also leaves us with something to really think about.
Oh, what's that?
Well, if a virus that evolved millions of years ago in bats and shrews can quietly jump to mice and then suddenly adapt to find the exact biological key to unlock human blood vessels, what other ancient viruses are quietly adapting to our rapidly changing world right now, just waiting to be discovered?
Oh man, that is exactly the question that keeps virologists awake at night.
Definitely something for all of us to mull over. Thank you for joining us on this journey today. Be sure to follow and subscribe to Deep Dive Lab for more fascinating science stories unfolding in real time. We'll catch you on the next Deep Dive.